Так, ну що, збираються? I'm very happy to introduce Taras Aleksik, who is one of the organizers of this school, and he will present his lecture today. And his research interests are in genomics, evolutionary genetics, and comparative genomics. And currently, his lab is advancing research in Ukraine to discover new genetic factors that affect the development and pathology of type 1 diabetes. And he's also working in the evolution and conservation of endangered species. And he is involved on studies focusing on genome assembly, variant analysis, and selection scans in wild and domesticated animals. And also, he, after collaboration 1,000 Genomes project in Puerto Rico, he organized and published population genomic studies in different parts of the world, and specifically in populations of Eastern Europe. So we are happy to give floor to Taras.
Thank you very much. Can you see my presentation?
Yes.
Okay. Hi, again, thank you very much for a nice presentation. I am very, very happy to be here and to be able to give a talk. As I said in the opening, Uzhhorod University is my alma mater. I'm very happy to participate this and organize this, and I only regret that I cannot be there in person because I love my city very, very much. But I'm not very, very far away. I'm a professor at the Oakland University in Michigan, and I'm also I have an appointment at biology department in Uzhhorod. However, right now I'm a U.S. Fulbright scholar in Krakow, Poland, associated with the Jagiellonian University, Silesian University of Technology, and Institute of Pharmacology of Polish Academy of Sciences. So I will be here for the next 8 months, and if you're passing through Krakow, please stop by, let me know. All right. What I will be talking about today is a general idea that it is important to understand human genetic diversity in its entirety, and if there are genomic gaps as in coverage and our understanding of genomic diversity, it in fact hurts us all and it hurts our health. I will start with the beginning of my field was the start of the Human Genome Project, which was probably the most important and most successful investment that United States has made in its history, because only in 10 years it had 100-fold, 100 times, 100-fold return of the investment. So Human Genome Project was about $3.5 billion. It came back with $300 billion within 10 years. That kind of investment you cannot even make on a Bitcoin. Okay, this is completely out of reach for most investments that were made. Now, the main goal of Human Genome Project was to create a map of 3 billion nucleotides of human genome, and basically make a map, a sequence. And the sequence was just basically a string of letters without any information. And more importantly, to our field, no information about the variation in human populations was made. It had to wait for several years. And as Svetlana said, I became part of the 1000 Genomes Project, which was a project that was organized worldwide, uh, uh, in starting in 4 countries and then spreading out to about 27, where uh, researchers got together to with two goals. One is to discover medically related variations that they could not discover by just looking at one human genome. And the other one is to explore genetic variation among different ethnic and geographic populations, which would allow them to ask questions about human evolution. Um, uh, in human evolution, in not only deep evolution as an evolution of human species, but more importantly, recent evolution of humans, evolution of in human populations. And as you know, the narrow definition of evolution is change of genetic frequency in a population over time. So what we want to know is from this data is what led, well, how different the populations are, what different variants are in populations, and what caused these differences between populations. And I would say there is another important aim. Um, because of the design of this project, is that this worldwide collaboration allowed to contribute to this research to all humankind. So everybody could contribute, and it will contribute to everyone. So not only select a few that lived in rich countries, such as in England or United States or Japan, would benefit from it. And this project basically started about 20 years ago, about 6 years after Human Genome Project, and resulted in two papers that at this point have about 30,000 citations. So if you look at my profile, it has more citations than anybody that you know, and it's only because I was part of this project. In this project, I coordinated one population. It's population of Puerto Rico. That's where I worked. PUR. It was 104 samples, and this really changed and really it changed the way people looked at that population, because all of a sudden there was all kinds of possibilities in studying Puerto Rican population in health, in evolution, in history, in demography, etc. So having this data that's available leads to benefits in the populations that study this. By the way, this map that you see here is from Wikipedia, but if you've stumbled on this map, it actually comes from my paper that talks about how 15 years ago, how different parts of the world deserve to have their genome. And the main idea of why that is that I believe in equitable research. Equitable is an English word that just means characterized by equity or fairness, just, and right, fair. Um, so equitable treatment for everyone, all citizens, all humans. These genomic advances that we are making in genomic studies should become accessible and beneficial to everyone. And this is very crucial for improving public health everywhere. Okay, so my view of genetic diversity is that it needs to be freed from corporate and government restrictions. It should be used by researchers to benefit of all. because it leads to understanding of human genetic diversity. And that is something that I will justify in this lecture. I will basically lead to you to understand why having access to data about genetic diversity is so important. And it's not only important from understanding who you are as a in a member of human species, right? But also that is important to public health, to medical science. It's important to your health, and it's important to economy and the well-being people around you. And as such, I will start talking about disease genomics. And if sometimes people think that evolution and disease genomics are two different things. And oftentimes people think about evolutionary biology as something completely removed from medicine, and as something that is focusing on Drosophila or C. elegans or perhaps on parrots in the Caribbean, my favorite topic that I talk about and work with, and maybe I'll give you talk about that later on. Or at least maybe finches, Darwin's finches in the Galapagos, right? Or maybe evolution of humans from chimpanzees. But there is, on the other hand, there are people in medical sciences that tend to ignore evolutionary approach and say, wow, those scientists, those PhDs in the universities, they really don't know what they're talking about. Here, we are in clinical applied medicine. We understand it better. We see cases in front of us. We don't need your advice. Well, that's not right on either sides, okay? And there is a science called evolutionary medicine, and big part of this is disease genomics. That the part that is crucial to understand how genetic and environmental factors shape human susceptibility to disease over time. In other words, why do we have diseases? Why are they distributed the way they are? Why somebody gets them and somebody doesn't? And what if in how this genetic component of disease, a host genetic component, human component to that, makes you more prone or less prone of getting it or surviving it. Okay, so some terminology before I start. In genomic science, we operate with a couple of things that I'm not sure if it was covered in the course yet. And generally, we discover these through genome scans or something called the whole genome sequencing. So whole genome sequencing is basically sequencing entire genomes. Um, meaning documenting every part of the genome, the entire sequence of the entire genome, all the chromosomes. We can do a cheaper alternative, which is focusing only on exomes, the whole exome sequencing. In the whole exome sequencing, we will focus only on genes, which is about 1.5% of the genome. Or we can do genotyping, but genotyping is this procedure that really makes it's really cheap now. But unlike sequencing, where nucleotides are all the nucleotides are read, genotyping determines states only a targeted set of genetic sites within the genome, and which means that before genotyping, you have to do sequencing to discover where the variants are. And if you discovered variants in the wrong population, then genotyping is, it might not be very well applied to your population, which is why sometimes genetic tests are from one population don't work in the other population. But we'll talk about it later. And you probably need to know this term called genome-wide association studies, or GWASs. And GWAS is a way of comparing diversity in genomic diversity across all the chromosomes in a population, which means aligning genomes of multiple individuals and looking at the differences and looking how these differences correlate with having or not having a disease. So on the right side, there is a scheme of a GWAS showing you have populations of cases and controls. You compare them to each other, and you see if a variant is present or absent. And in this case, cases have, like people with the disease, have that red A in the sequence, and the controls have blue gosh, I am completely blind, a blue T in it. So cases have A, controls have T. Well, A must be associated with a disease. That doesn't mean that A causes a disease, but it might be in close or related to or linked to a variant that causes a disease, but it makes our understanding of disease a little bit better. And other things that we need to know here is some types of genomic variants that we're looking at. So you probably know this, but if you don't, we operate with things like SNPs, single nucleotide polymorphisms, which are either substitutions, uh, or it could be insertions or deletions if they're just one. Then we talk about indels. Again, indels could be one nucleotide or many. We can talk about CNVs, or copy number variants, which is repeats of, let's say, part of the gene or the whole entire gene, and structural variants, or SVs. So these are all types of things that we look for in the genome. And then once we know them, we can compare them between individuals in order to say if one of these is associated with a disease. Now, diseases, of course, we have to classify as well. So I don't know how much of this you know, so I will repeat it. Diseases could be monogenic, or simple, or Mendelian. These are caused by mutation in a single gene. It seems to be pretty simple. One gene, right? One mutation, one disease. And because of that, they follow Mendelian inheritance, and you can have it either autosomal dominant or recessive or X-linked, etc. Examples, we're going to look at some of these examples, like cystic fibrosis or Huntington disease, as opposed to polygenic, and these are complex diseases. They result from combined influence of multiple genes, and often involve environmental factors. And they do not follow Mendelian inheritance patterns because they're a lot more complex. So we call them polygenic or complex diseases. An example of that would be type 2 diabetes, and actually also type 1 diabetes, that involve genes related to insulin production and glucose metabolism. And they're influenced by lifestyle factors. So some people will get it, some people won't. So again, simple Mendelian trait disease is just like Mendel's peas, right? Would they follow Mendelian inheritance patterns? They exhibit varying degrees of penetrance or probability the gene will have the phenotypic expression. And they often, as a disease, present with a specific set of symptoms and well-defined symptoms. So you have them, you will be recognized with them. Right? Think about things like Down syndrome. You can basically look at the person and say, well, they have a Down syndrome. And common diseases are often polygenic. They have an additive component, which means they are additions of different genes together. So you may have some of them expressing, or some more expressing in other individuals, and very many of them expressing in the other individuals. So you adding up different mutations, and often the more you have, the stronger case you have, but they might not be the same ones. And they also have this environmental component. And today, a way of assessing the risk that you have is not based on having or not having a disease allele, rather than having a polygenic risk score where you have combinations of many alleles together, right? Now, I will not talk about this today. We will just look at simple diseases. It's enough to demonstrate evolutionary factors in them. But for complex diseases, come to my class on Bioinfo UA. These lectures we already posted online, and you can simply repeat the course. Part of it is on complex diseases. All right, so let's go back to the simple diseases, and let's talk about one of the most famous examples, which is sickle cell anemia. You remember this one, right? Sickle cell anemia is a condition where one of the a very simple a mutation causes a change from glutamine to valine, and that actually changes a shape of hemoglobin, and that results in a different shape of erythrocytes, and they become sickle-shaped, serpovidny, sickle-shaped, and this causes all kinds of problems because they get stuck in your capillary. Now, this is a Mendelian disease. It actually follows Mendelian inheritance. Sorry, I thought I actually had it here. Yeah, it is a Mendelian disease. It follows a Mendelian inheritance. In order to get sickle cell anemia, you need to get it from your both parents. The allele from both parents, or you can actually have a heterozygote, which in heterozygote does not have a sickle cell anemia. We call this type of inheritance codominant. Sorry, we call it's a dominant inheritance, but the sickle cell, we call it codominant because, and I didn't tell you why it's not codominant, because it's only an environment that I don't describe here. But in this case, sickle cell anemia comes from people having both alleles, and you don't have sickle cell anemia in either heterozygous or homozygote. Now, it is codominant for something else, for some other response, but I will get to that later. From this point of view, it is actually dominant. All right, so if you get sickle cell anemia, you obviously have not a very comfortable life. But very luckily now, this was actually the first ever disease to be associated with a mutation that happened about 50 years ago. And because it's a simple thing, right, it actually means that you can be targeted with a therapy because you know exactly what the mechanism is. You just target the mechanism of this mutation, and you either knock it down or make a replacement, and you can cure it. So in fact, FDA in the United States has finally approved a treatment for sickle cell anemia where they, in fact, replace this mutation, knock it out, and are able to cure people from it by literally genetic therapy. Okay, now, a lot of these monogenic diseases they're simple diseases. You think about them as something you don't see a lot, right? You think about them as rare, while polygenic diseases are pretty common. And we will actually look why that is. But I want to say that these Mendelian diseases, in fact, they may be rare, every one of them, it may be rare, but as combined, they actually very common. So there are about 6,000 rare diseases that are simple genetic diseases that affect about 30 million people in European Union, and that costs about $700 billion a year. So understanding how this works is very important, because it has a huge impact on public health, and it's a big part of your economy. A lot of part of the economy of most countries goes for healthcare, and therefore understanding this is probably paramount. All right, so what can cause distribution of these diseases in population? Well, of course, what operates inside of populations are the evolutionary forces. See, and understanding the evolutionary background provides important context to studying these simple diseases. Most of them are caused by historic mutations, and these mutations either were neutral, they didn't have very much impact for most of the time, And then they may have turned deleterious. Deleterious means disease-causing, or advantageous, which means protective. Okay, and the genetic variation in the population can be studied with good old evolutionary mechanisms, forces. As you know, these are mutation, selection, gene flow or migration, and genetic drift. And there's one more here that's usually not mentioned, and it's non-random mating, and it's a very, very important force. But I will not talk about it in this lecture. All right. So the first force that we'll talk about is mutation, and people think of evolution as mutation all the time, but you probably already know that mutation is the weakest evolutionary force there is. However, it does cause genetic disease. So an example of mutation caused by this is very famous, Huntington disease. That's caused by expansion of CAG repeat in HTT gene, Huntington gene, on chromosome 4. And so what happens there is that the longer this repeat becomes, the more likely it is to be extended. So if you have 41 repeats, then well, if you have like 7 repeats in this gene, then it's not likely to be expanded in your children to 9. But if you have 41, it's very likely that it's going to be expanded. So the longer it is, the more unstable it is. So that's called genetic instability. And then what increases mutation rate. And so new generation will inherit more CAG repeats than the previous one. And so in the population where you already have many repeats, it is likely to get a Huntington disease. And Huntington disease is something then if you have over 40 repeats, then your neurons get degenerated, and it's actually pretty bad because you get this disease in the age 25, 26, when you're already formed individual, and then all of a sudden your brain starts falling apart. Now, it may not have affected your reproduction as much as if it was very, very early, but you still only have very limited part of reproduction. And in human species, you also need to take care of your children, as I very well know with my children. And so if I didn't take care of them after age 25, they probably, not do very well. So it does have impact on fitness. All right, so you may have heard the story. The very famous researcher, Nancy Wexler. At the time, she was University of Michigan. Now she's at Columbia. She herself was from a family with Huntington disease, and she noticed one interesting thing that Huntington disease is distributed mostly in the places in the north, in Europeans, but there's one place, specifically one dot in South America, in a place called Barranquitas on Lake Maracaibo in Venezuela. And in this place, there was an unusual number of people with HTT. So she actually went there, and you can see a picture of her drawing the pedigrees of families that live there, and she was able to trace the mutation to a single founder that arrived there from Europe about 200 years ago. So this mutation effect was in fact amplified by something called founder effect. Now we know founder effect is a type of genetic drift, and genetic drift is a very strong evolutionary force. So genetic drift as an evolutionary force, we usually think about it as something that acts in smaller populations. So as a conservation biologist, I think about cheetahs, which I wrote papers on, and parrots and solenodons and et cetera, elephants, rhinos, species that live in small populations. And what happens in small populations over time, because of drift, it will lose variation, but it loses variation in the results and the fixations of alleles that might not be advantageous. So if a population is small, the danger is that in these populations you will arrive to everybody will be the same, and everybody will also have an allele that's not very good for your fitness. So yeah, on the one hand, we think about this mechanism. I think about this mechanism in the Caribbean islands when we'll look at parrot speciation, and a flock of parrots goes to an island, and after 1 million years, it becomes a different species. Right. On the other hand, you could probably know examples of people going to an island and at some point running out of genetic diversity and developing a certain condition that's not common to anywhere else in the world. Like one of them is a case for retinitis pigmentosa, which is a hereditary blindness that occurs in higher frequencies on the island Tristan da Cunha. Now, Tristan da Cunha was founded by about 15 people that came from Great Britain in 1814. And at some point, most of them, because of this drift and founder effect, were ended up with hereditary blindness. Everywhere else in the world, this is rare, but in Tristan da Cunha is very common. And there are populations like that that are isolated and arrive to different type of disease gene mutation fixations that are higher in these populations than anywhere else in the world. And in fact, that allows us to study these diseases in these populations. We call them population isolates. Population isolates are people that tend to have endogamy, so they will only marry among each other and stay in the closed knit groups. And then because of that, the effective population size, which is size of reproductive population, is smaller. And they often run into these develop the higher frequencies of these diseases. So there's a list of these here. The Amish, I don't know if you've heard of Amish. These are religious communities in United States that arrived from Europe 200 to 300 years ago and still live in endogamous communities. The Hutterites, the people, some Jewish communities that were isolated that developed some of this as well, and then also some island populations like the Sardinians and Marquita Island people. So now, of course, when the drift is strong, it will get rid of mutations. But if the drift is not strong, the population is larger, then mutations will accumulate. And humans will accumulate mutations because mutations just happen. They do happen randomly, whether they can happen anywhere. Of course, whether they stay or not depends on the size of their effect. The size of the effect that will bring in selection, in this case, negative selection that's strong or not. So if the mutation has a big effect, then it will invoke strong selection against it. Okay, so here's an example of very, very strong effect. Progeria. You probably heard of progeria. These are people that are age very, very fast. At 5-year-old, they look like they're 105-year-old, right? And this is a rare genetic disorder that's caused by sporadic mutation in one gene, which is known as lamin A, LMNA, which makes a protein that's needed to hold the nucleus together. Of course, if the nucleus is not holding together, everything's falling apart. Now, if you look at the distribution of progeria mutation around the world, this is a distribution, right, of about 400 cases So it happens at about 1 in every 4 million live births worldwide. So about 400 cases are currently alive with progeria. They live to a maximum of 15 years, 16 years. That was a very famous, very outspoken progeria patient that died and wrote a book about, too old for her age. Um, and the problem is with these mutations is that if you get it, you will not survive. And of course, you will never live to have your own children. So selection practically eliminates all of these mutations. So it pops out and it's deleterious, completely deleterious. Okay, let's look at another one. Seems to be pretty deleterious to me, right? Cystic fibrosis. You may have, or CF, you probably heard about this as well. This is the disease that causes, that changes how much salt moves in and out of your cells, and it causes salty sweat. That's one thing, one symptom. And and inside of the digestive system and lungs, it creates a lot of sticky mucus that makes it hard to breathe. So at some point, it's hard to breathe, and at some point they get these lungs get infected, and these people that have cystic fibrosis, they normally in the past did not survive past 15 or 16 years old. Now, this is caused by many mutations inside of this gene that, and of course, the effect of it is different. Is some of them actually cut off part of the gene, some of them just substitutions. So the effect of each mutation is different, but in either case, if it leads to cystic fibrosis, these this this should invoke very strong response, right? Strong effect. Um, however, if you look around the world, you will realize there are about 40,000 people with cystic fibrosis just in USA, and another 60,000 people living in Europe. That's a lot more than progeria, right? And now, cystic fibrosis is a dominant disease, so you could probably still survive with it in the heterozygote. But there's a hypothesis that the reason why this disease is maintained at higher frequencies in these populations is because in the past, the heterozygote was protective against cholera. So if you have a homozygote, you will have cystic fibrosis, but heterozygote was actually good for you. You will survive infectious disease. Okay, so it looks like this case is not caused by mutation alone, but it's caused distribution of this disease is an intersection of mutation interacting with natural selection. Okay, there's a that was a past selection that maintained mutation, right? Maintained disadvantageous mutation. Now, here's another example that's kind of the opposite. So here is a mutation that was very common in Iceland in cystatin C gene 200 years ago. Very common mutation. Frequency of this mutation was, somewhere along 10%. Right? Um, that's that's pretty high frequency. 10%, it's pretty high frequency. And then towards the middle of 20th century, this mutation has disappeared, started to disappear. And it's now hovering on 0.5%. So only if you are heterozygote, you'll have it, according to Hardy-Weinberg. Now, Icelanders, of course, a genotype and sequence everywhere in the country. They have a very famous consortium, decode, and they um, were able to look inside of their records, in the church records and legal records, to see who has had these diseases, this disease in the past. Sorry, who had carried this mutation in the past. So they were able to go through family pedigrees, and trace back to 1800s who had or had not in the past. And what they also knew from these records is how long did these people live, right? So whether they lived, 50 years or 20 years, 60 years, etc. So that's what you see on the graph on the right. You see the graph which shows that the life expectancy of people with this mutation has decreased from 80 years on average 200 years ago to 20 years in 1920. Now, what happened, you had these mutations accumulating in the population, and they did not have any effect because the environment in Iceland was not such that this mutation would cause harm. But as the Iceland was reached by European foods, so they used to only eat fish and vegetables, right? And they started getting starchy foods from Europe. All of a sudden, this mutation has expressed in the way that became deleterious. So people that carried this mutation died, and as they died, they would not pass on their mutation to the next generation, and the frequency of this allele in the population collapsed. So there is no more cystatin C deleterious allele anymore. All the people that had it died from stroke. All right, so these are these are the take back message about this. These deadly genetic diseases like progeria should be rare because they will be eliminated, but selection can maintain disease alleles, right? And we go back to our first example that we had with sickle cell disease, and we know that it was maintained, and this is why I started debate with myself. It's not actually a dominant disease. It's actually co-dominant disease because on one hand, it will cause sickle cell anemia, but on the other hand, it actually, if you don't have sickle cell anemia, if you have a homozygote for the other allele, you will actually die from malaria. And the only way to survive is to have in these Central African environments is to have both alleles. But unfortunately, because of Mendelian inheritance, heterozygote has to be segregated. So you cannot inherit it. You only inherit alleles that will combine in you from your parents. And therefore, the population will carry this heterozygote because it's good for population, but it might not be good for an individual that did not inherit it. And once you move somewhere else, it's actually bad for you. And in fact, migration, also an evolutionary force, brought these two alleles to the United States where there is no malaria, but sickle cell anemia became a big burden, and it's associated with African fragments of the genome. So if you have a sickle cell anemia, it probably came with this piece that was really good for you when your ancestors survived with this in Africa, but today you might get sickle cell anemia. And of course, this is a consequence of a terrible page in human history called the slave trade, where Africans were brought in as slaves first to the Caribbean, um, and then to the United States. And then they were used as a slave labor on either sugarcane, tobacco, or cotton plantations. Anyhow, this in this case, something that was an adaptation to your survival in one place, now it's migrated, and now you have migration brought it to another place, and changed the frequency of alleles in this place where you perhaps not needed anymore, and it became a disease. Now, that makes us think that a lot of genetic diversity that has to do with disease actually might be something that was a human adaptation in the past, but today it's not needed. So you think about many places in the world where a specific adaptation has occurred. For example, you have adaptation to cold weather or to fat foods or to again to malaria or to drinking milk or to living in the high elevations. Um, Richard Lewontin, a very respected American evolutionary biology, famously argued that because a vast majority of human genetic variation is within population and not between them, there's not very much difference between us. And it is very true. There is not very much difference between us. We are still one human species. However, there are differences between us that are very, very informative. And it's like looking for a needle in a haystack. Finding each one of them is a big, big reward. Understanding how human species has solved one of these problems during its evolution, right, is a very valuable lesson that we can learn. And this is what we call an adaptationalist agenda. We'll return to that again. So one of the things that this does is it results in some variants to be specific to one population, but not in the others. A very famous example of this is something called a Duffy null allele locus, and it's absolutely exclusive to Africans. You cannot find it anywhere else. And this was a reaction to, again, malaria in Africa. So people that have this are resistant to a specific malarial parasite. All right. So if you have alleles like this, you can tell that the person has a similarity. Now we call it genetic similarity, but has a piece of chromosome that's similar um, to the one that's derived from Africa. Of course, it's similar, not necessarily it, because it could be caused by a random mutation in your lineage. So you can't actually say that for sure until you establish the ancestry. Okay, but if you have lots of these, you can in fact map your genome and figure out where different parts of your genome have been, right? So as we know, as two parents with different ancestries are mixing, what happens, what happens during meiosis is a process of recombination, which is the exchange of the little pieces between chromosomes. In fact, it should be should happen at least 3 to 5 times per chromosome per generation. So it happens a lot. And as these pieces are infiltrating into your population, they contribute to a variety of ancestries along the chromosome in you. So what we see here is the red population is adding members to a yellow population, and what you have there is increasingly smaller pieces of what we call admixture. So this is a this is called by crossing over, and the result of it is an admixture. So migration leads to chromosome being composites of chromosomes of your ancestors. Okay, and so what we can do is we can pick these ancestry markers, or we call them AIMs, ancestry informative markers, that have come to fixation in different populations because of its evolutionary history. And we can map the chromosome. So let's say in this chromosome we have 2 markers, so we're pretty sure that it came from Africa. This one again, it has a marker from Africa, and this one again I have from Africa, and this one has one from Europe, right? So we can paint chromosomes, and this is called local ancestry, paint chromosome locally and identify fragments that came from one place or another. All right. And with this, we can now start asking questions like, what happened to this fragment? Like the fragment that was bad for you, that was had that ancestry, it would probably be removed by selection. But the fragment that was good for you for your survival, it will increase in frequency. And this is actually one of the bases of finding natural selection in the human genome. And if you want to learn more about this, I actually wrote a whole big review a while ago, and it's been cited multiple times. And I would urge you to look this up. It's in 3 or 4 textbooks now. So anyway, so when I was writing this paper, I actually noticed there was there was this graph that I used, and this is the part of chromosome in of Puerto Ricans. Puerto Rico is where I lived at the time, and this is chromosome 6. What it does have here is too much African ancestry, too many of these African pieces of chromosomes as compared to Europeans in chromosome 6. And now knowing this, it actually kind of made me think about what it means. What it means that every time you have a new chromosome, a new baby, and it has a European piece of chromosome in there, it's not good to survive in Puerto Rico. If it has an African piece, it's actually good to survive in Puerto Rico. And so over time, people that live in Puerto Rico, they're admixed, partially 60% of them, their genome is European, 25 African, 15 Native American. So what happens here is there's too much African. It's not 25% African, it's 40% African in this particular part of the genome. And this part of the genome, chromosome 6, is HLA region, which is responsible for immunity. So immunity is important, and it's been selected. And in African immunity is good to survive in Puerto Rico. For example, this other place here, this is a study that we did on kidney disease, and these are people from hospitals that have been diagnosed with renal a kidney failure. So people that have kidney problem, we sequence them and we identify their ancestry, and their ancestry is even European and African ancestries, even along all the chromosomes is about the same. About 80 to 20, 80 to 20, 80 to 20%. It was African to European. And then on chromosome 22, there's a piece that's always only African. So that if you have only African piece on chromosome 22, and you have sorry, if you are an African American admitted to a hospital, you will have an African piece on the chromosome 22.
Taras,
Okay. Yeah.
we have limited time, so you have 6 minutes to finish and for discussion.
It's okay. I will finish. You don't know how many slides I have, so okay. So that and I'm watching my time, but now I added another minute for this stop. Okay, so when we look at that and said, okay, here's a piece that's enriched in Africans. So what is in there? What's in this piece? Now underneath this piece was about 32 genes, and one of these genes called MYH9 was known in renal disease. Okay, and it was known in renal disease, and because of that, doctor said, okay, this must be the gene. Now, when I was a kid growing up in Ukraine, I was reading kids' books. You probably read these books too, right? Tomek on African continent, right? Or Tomek in the Wild West, whatever that was, by Arthur Shklarski. And I remember a moment where the expedition goes to Africa and they find people that are asleep. And they call this a tsetse fly, and bit people, and they have a sleeping sickness. And I start looking for genetic determinants of sleeping sickness. The gene that came up was APOL1. And guess what? It was one of those 32 genes. So I started arguing that, hey, this is not MYH9. It has to be APOL1. But I was overruled in one paper although I did come up with a Nature Genetics paper. But because of my argument, when people actually looked at APOL1, they included me into the study, and I actually got a science paper from it. The point is that when you look at these medical records, it is important to understand that nothing in biology makes sense except in light of evolution, as famously said by Theodosius Dobzhansky, who is a great Ukrainian American evolutionary biologist and author of the Modern Synthesis. So, well, I ended up writing another paper about this. But my inspiration was the books I read as a kid. So it's important to understand, but it's important to expect that this discovery would not be impossible if we didn't understand the global distribution of MYH9 and APOL1 alleles to understand that APOL1 allele is common in Africa, only in Africa, which shows the sign of the old adaptation. So, wrapping this up, understanding disease mutations is impossible if you don't understand global distribution of genomic diversity. And where are we with understanding genetic diversity? Well, we're in a tough place. Even in Europe, we have blank spots of genomic diversity. And then this is a paper we published about 2 years ago with Walter. And this is a paper Walter is submitting about now, right, to Cell. And we still have huge missing pieces in genomic diversity that are not explained. And these pieces are missing in places that are where in countries that are being developed where the resources are not available or people are poor. So in fact, it results in unequitable research. So it is important for us to understand the full picture where we can go from distribution of genetic diversity looks like this to this distribution of genetic diversity looks like this. And now we can kind of, sail and discover things. So what do we do here today? Well, we do locally. Well, we started a Genome Ukraine project, which actually added to population genetic structure in Europe, put the piece in there, right? This is Ukrainians, right? These are Ukrainians and Polish, and that's a Russians. Okay, just so we know. This contributed a lot of novel SNPs that can be now looked for by the doctors and contributed to GNOMAD database that can be where they can be searched so we can discover them. And then we moved on to even larger initiatives like type 1D exomes in Ukraine, which is a huge program that's run by Uzhhorod University, where you are right now. Where we collect samples from all over Ukraine with people with diabetes and people without. At this point, we're at about 11,000 samples. And we're analyzing variants so that we can then look at them and understand them. So we can tell that we're filling in the very specific part in the genetic map. And in this genetic map, we're going to look for a specific determinant of type 1 diabetes in Ukraine. So this is actually a graph that nobody has seen before. And it shows there's some local determinants of type 1 diabetes in Ukrainians. So as the result of this is the program at Uzhhorod University in genomics, and the school that you have there is actually one of the reasons school is there is because of this project. This project is now expanding to other places in Eastern Europe. That's why I'm in Poland. And because of this, we became part of Genome Europe. So we're representative of Ukraine in the Genome Europe, which is not on this picture until now, because in the kickoff sailing of Genome Europe, we were already present. So we're going to be collaborating with this huge project, which will fill in the missing pieces in genome map of Europe. All right, so that is all thanks to our efforts, and I would just like to admit these people that some of them are in the classroom, thanking them for what they have done and contributed. So if you have questions, if you don't, you can just reach me by email. Thank you. Thank you, Valery. I know you're part of it too.
Very such a good talk. I think we can have only one question, and other questions people can ask in chat or by mail.
Yeah.
I saw this hand first.
Go ahead.
Okay, so thank you so much for the opportunity to ask you one question. I basically would like to delve deeper into cancer because we can consider this process, I mean, tumor genesis, as a form of somatic evolution. And you also have mentioned that in terms of statistic process, it can be like some former adaptation to like new environments. So is there any considerations related to cancer by this point? I mean, can we consider cancer as one of the forms of adaptations to some circumstances?
Thank you for a very good and very relevant question. I did not cover this part of disease genomics in this presentation, but I could have, right? So that's just another part of evolutionary medicine. Of course, cancer is an evolutionary disease. Cancer cells are they are evolving, evading our selective pressures. They found ways of um, adapting to the environment inside of your body. They're basically rogue cells that are, they found a way of evading regulation. It's also a great example to emphasize that evolution is blind because these cells are out there to survive themselves, but what they will do is to kill the host, or you, right? And in the end of their evolution will be for nothing. They'll basically crash the organism while trying to get all the resources to them. So yes, cancer cells have absolutely every hallmark of evolutionary process. Cancer is an evolutionary disease, and now lots of evolutionary knowledge is co-opted into understanding how cancer develops and how we can develop therapies that can stop cancer progression. Okay. It's not only about cancer. So if you think about cell evolution inside of the body, it's about aging. Aging is, again, you can build phylogenies of cells. Now people sequence cells inside of blood and figure out phylogenies. You can apply evolutionary principles everywhere. The time has passed when evolution has been only a theoretical science. It's a very applied and practical science today.
Thank you so much for that.
Thank you very much. And unfortunately, we don't have time for more questions. Okay. And now I want to introduce you, our next speaker. It's my pleasure to introduce Alan Bergland. He is working at the University of Virginia, and his research focuses on the genetic basis of rapid adaptation to seasonal and spatial variation in selection in Drosophila and Daphnia. He combined fieldwork, large-scale phenotype and genetics and genomics to identify the genes and traits underlying local adaptation and to study evolutionary forces that maintain genetic variation within species. He started his education at the University of Oregon and completed a PhD at Brown University. He had some postdocs, and now he is a professor at the University of Virginia. So, Alan, the floor is yours.
Okay, thank you very much. Thank you very much for the kind introduction and for the invitation to come and share with you some of the work that we've been doing studying seasonal adaptational and seasonally varying selection in Drosophila. And I want to start just by motivating the bigger questions of the work that we do. So first off, everything is good with the projector? We're okay? All right, so the big question that that motivates my work is understanding or asking why is there genetic variation in populations? And this is, of course, a central question to evolutionary biology and all of biology, as Taras has so nicely laid out in his talk. What I'm particularly interested in is understanding how environmental variation across the species range or through time can act as a force that promotes genetic variation in populations and promotes functional genetic variation in populations. And so we study how temporal and spatial fluctuations in selection pressure maintain variation underlying fitness-related traits. And we do this work in two groups of organisms. We work on Drosophila flies, mostly Drosophila melanogaster, but some of its close relatives, as well as Daphnia, and we focus on primarily Daphnia pulex and on European Daphnia pulex. But we work on these organisms for a couple of reasons. One is that they are both broadly distributed throughout temperate environments and tropical environments. So they're widely distributed and experience a wide variety of selective pressors across their range. Both of these organisms also have a rapid generation time, maybe 5 to 10 days. And so over the course of the growing season, there are multiple generations per year, and each generation experiences a different aspect of the environment, right? That's changes in aspects of the abiotic environment as well as biotic interactions, right? Things like parasites and predators, as well as temperature, nutrition, resources. And over that period of time, over the course of the growing season, these species change in population size dramatically, sort of adding additional selective pressures to the populations. So in my lab, we try to study the evolutionary processes across spatial gradients and time. And we do that using a variety of different tools and approaches. We try to understand the genetic architecture of the traits and behaviors that are associated with fitness using GWAS and QTL mapping. We study the evolutionary dynamics of alleles and populations through population genomic inference. Together, that work tries to infer the environmental drivers of evolution, and then we use tools and methods we develop and also benefit from like a really nice sort of community of scientists around the world, which is how I know Sveta, as well as citizen-based science efforts. Before I begin, I just want to acknowledge the folks in my lab that have contributed to this work, as well as the collaborations and that I have and funding agencies. So today I want to talk about seasonal adaptation in Drosophila flies. I'm going to address three basic questions. One is seasonal adaptation, a general feature of fly populations. What are some of the environmental drivers that could be promoting seasonal adaptation? And then what are the genes and loci that underlie this phenomenon? So I want to start by defining how I think about seasonal adaptation. And I think there's two ways that you can think about seasonal adaptation. One is seasonal adaptation as a form of phenotypic plasticity. And this occurs in organisms that have long generation times or have the capacity to migrate long distances. So these are things like bears that hibernate, butterflies that migrate, but also us, right? We put on jackets when it's cold and we put on shorts when it's warm. On the other hand, if you're a short-lived organism, you can think of seasonal adaptation as adaptive tracking, where you can have different genotypes that are specialized to different periods of time, summer or adapted. And over the course of the growing season, over multiple generations, the frequency of those different alleles will fluctuate in frequency through time. So Drosophila are an excellent model system to study seasonal adaptive tracking. They have these huge population sizes globally, and also locally, right? So selection is very efficient. They have multiple generations per year. The populations grow very rapidly. They're distributed around the world, and then they're of course fairly easy to collect. Flies live in orchards, right? These places that have strong seasonal changes in their environment. And when we think about the general, population dynamics of melanogaster in an orchard, this is a sort of a cartoon we can come up with where fly populations over the course of the growing season, sort of broadly defined, increase in size. And then there's this period of overwintering where fly populations maybe survive as adults, but then there's periods of unfavorable seasons, maybe before there's resources where they might eke out a few generations, but life isn't very, it's not very resource abundant. We know and we can estimate winter population bottlenecks in these populations using genomic data. And so we can show that there's a strong reduction in population size through time over the course of the winter, upwards of 95%. But nonetheless, populations are resident, right? So from year to year, the same genetic sort of population exists in the same spot. And that locally, there's fairly small effective population size. And we were able to arrive at this conclusion by studying patterns of allele frequency change through time in flies. And this is one example. So these are samples of flies collected across Europe and North America over multiple years and oftentimes multiple times per year. And this is a principal component representation of genome space. And you can see that different populations cluster together through time, but then there's change through time as well. And we can use the magnitude of change in frequency through time over winters to estimate these population sizes. So for example, we have estimates of FST, which is a metric of population differentiation, between samples collected within a season or samples collected over winters. And you can see that the overwintering FST is larger than the within season FST and therefore conclude that there is a winter bottleneck. And you can use the extent of FST to make these estimates of effective population size. So if you go out and sample populations at different times of year, you can get these estimates of allele frequencies, but you can also measure phenotypes, right? And you can take those flies, bring them back into the lab and rear them in a common garden for multiple generations, and therefore be able to test genetic differences in phenotypes of flies collected at different times of year as a way to reflect adaptation to temporally varying environments. This is work from a collaborator of mine, Paul Schmidt, and his lab, looking at phenotypes in flies that were collected at different times of year, but reared in a common garden. And you can see that there's changes in things like starvation tolerance, the pigmentation of flies, so how darkly pigmented they are, as well as the genetic propensity to enter diapause, which is an insect form of hibernation. So phenotypes change through time, and what we want to, and we can sort of generally understand the change in phenotypes through time, the evolution of phenotypes through time, as reflecting a life history trade-off between somatic maintenance, right, which is going to be favored in the winter, as well as we think maybe more northern populations, or allocation more towards reproductive output, which is going to be more favored during the summer or southern populations. So if we believe that there is phenotypic differentiation through time, there must be alleles that underlie that genetically based phenotypic differentiation. And in principle, an allele that could underlie this sort of seasonal adaptation might have a trajectory that looks like this. So we have a red line and a blue line representing the summer and the winter adapted alleles. If you go out in the spring and sample flies, those flies are the descendants of those individuals that survived winter. And so we would expect the winter allele to be at high frequency and the summer favored allele to be at low frequency. And then if you go out in the fall, at the end of the growing season, the summer adapted allele in principle should rise to high frequency because it's associated with increased reproductive fitness, whereas the winter associated allele would not rise to high frequency. And so we would expect over multiple years, this zigzag pattern of allele frequency change through time. So this is the work that I did as a postdoc using samples collected in Pennsylvania in 2009, 10, and 11. We perform what's called pool-seq, which is where you take a sample of flies and you grind them up in mass and extract DNA, and then sequence the pool with Illumina sequencing, and that lets you estimate allele frequencies genome-wide. So in this plot, each thin line is a single allele, a single polymorphism. And these are the polymorphisms that we had identified as fluctuating in frequency through time. Now, there's about 1,700 polymorphisms that we identified in this study, and we don't think that all of them are independently responding to selection, but we think that some subset of them are, and the rest of them are sort of being dragged along via linkage. We wanted to then understand whether seasonal adaptation is a general feature of fly populations sampled around the world. And so in 2012, we organized a meeting at a Evolutionary Synthesis Center in the United States, where we invited colleagues from around the world to come and talk about how to sort of generate these sampling efforts over broader scales. We convinced several of our colleagues to sample flies near their home institutions, and so these green circles represent places where we have 1 year of samples, spring-fall samples. And so we were able to use these to then ask whether seasonal evolution was sort of observed in multiple populations. And the way we assessed this was that we built a gene environment association model where we modeled allele frequencies as a function of season for every single SNP. That model gives you a p-value, and then you can look at the distribution of p-values across the genome. And that's what we're looking at here in the red line. So this is sort of like a histogram of p-values, but on a log scale. And the red line is the real ordering of the spring-fall labels. And then the black line are 100 permutations where we flipped around the spring-fall labels. And what you can observe is that there are more seasonal SNPs with low seasonal p-values than you expect via permutation. And that is evidence that there is seasonal evolution happening in multiple populations. On average, the change in allele frequencies between spring and fall was on the order of about 5%, a difference in allele frequency. So this is relatively substantial change in allele frequency over a fairly short period of time. We've since extended this sampling effort through a wonderful collaboration with DROS-EU, which is the Drosophila Evolutionary Biology Consortium. Collectively, we as a community and as independent scientists have been sampling fly populations around the world. And so we have generated this data set called DEST, Drosophila Evolution in Space and Time. And this is a resource that collects all of that population genomic data. The latest version of this project has been spearheaded by a former postdoc in my lab, Joaquin Nunez. And you can see that we have samples collected around North America, Europe, South America, Africa, Australia, and over a long period of time. This data set collectively represents over 45,000 flies in total that have been collectively sequenced by this consortium efforts to over 30,000x coverage. And it's really a wonderful resource. There's a well-documented pipeline. There's a ton of curated metadata for each sample, and all of the data are available online. And you're welcome to download and to start to study it if you're interested in trying to study patterns of evolution through space and time. Flies. Okay. So we wanted to use this data set as a way to ask this question again, right? Is seasonal adaptation a general feature? The first time we asked it, the samples that we had used primarily came from North America. And so we wanted to ask whether in a set of samples that were exclusively found in Europe, whether we saw the same pattern of seasonal evolution. So we identified a set of populations where we had spring and fall samples that were sort of evenly distributed in different parts of the world. This is a data set that we ended up using. We did this very same analysis where we tried to identify allele frequency change through time. And we observed, we observed that there are more seasonal in North America than there are, or excuse me, in Europe than we expect by chance. Okay, so we're able to then answer again this question that seasonal adaptation is a general feature of fly populations. Okay, so next we want to ask, what are the potential drivers of seasonal adaptation in flies? There's a lot of ways that we can go about answering this question, and I want to just sort of focus on one of the ways that we have done this in the past. And this is an approach where we're utilizing this data set of 20 paired spring-fall samples collected primarily in North America and a little bit in Europe. And in this analysis, what we first did is what's called a leave one out analysis. And here we have our 20 paired spring-fall samples. And what we are doing is we're dropping one of those samples, and we can define the remaining 19 as a discovery set where we're trying to identify seasonal alleles. And in the test set, we can ask whether allele frequency change at a particular SNP changes in the same direction as the remaining 19. And then we can repeat this process sequentially, dropping each of the samples. And we can then look at the output of this in the following way. So this is one of these dropped samples. What we define is what we call the joint significance quantile. And so this is a threshold. We take the, for example, top 1% SNPs in the discovery set and the top 1% SNPs in the test set. And we can ask for those SNPs, what fraction of them are changing in a concordant direction, right? The same change in allele frequencies. And for example, this particular population, as you look at SNPs that are more stringently significant, there is an increase in concordance. So we would say that this particular test population is concordant with the remaining 19. We can do this for all 20 of the populations. And what you can see is that for 16 of those populations, there's a strong signal of concordance. But for 4 of them, they have a signal of discordance. And in essence, there are 4 populations that show a reversal. And by reversal, I mean that the spring allele frequencies look like the fall allele frequencies, and the fall allele frequencies look like the spring allele frequencies. And so we hypothesize that if temperature is a driver of seasonal adaptation, then we might expect these populations that show reversals to either have evidence of a warm spring or a cold fall. And so we were able to test this by extracting sort of daily temperature data for the samples in the weeks prior to sampling for all of these different samples. And we built a series of models where we calculated the number of days when the spring maximum temperature was above a threshold and the number of days when the fall minimum temperature was below a threshold. And we then define for each population what we call a prediction score. And the prediction score, you can see, is sort of related to the strength of concordance. And it's essentially the slope of these functions. And we're trying to explain variability in these prediction scores. And if you know the number of days when the spring maximum temperature is above about 32 degrees Celsius, or the number of days when the fall minimum temperature is below about 5 degrees Celsius, you can explain upwards of 70% of the variability in these prediction scores. And so these temperatures sort of align with some of the general kind of thermal tolerances of flies, at least into a certain approximation. And so this is a good indication that temperature in the weeks prior to sampling can predict allele frequency change across spatial gradients. This isn't the first or the last time that we've observed that short-term changes in temperature can drive allele frequencies. In an earlier paper, we showed that that frost events can actually sort of make alleles move in a more winterly direction. And then in a more recent paper, we can show that evidence in evidence that temperature in the 2 weeks prior to sampling is a good predictor of allele frequency change. So short-term fluctuations in temperature are correlated with allele frequencies and are potentially a causal selective agent or could be driving other environmental factors. So finally, we want to understand what the genetic loci and the phenotypes underlying seasonal adaptation are. And to do this, we utilized a set of samples that were collected near my home at the University of Virginia. So the University of Virginia is located back here, and within a pretty short distance is an apple orchard where you can go and pick apples and have donuts and things like that. And so when I moved to Virginia in 2016, we started extensively sampling this orchard every 2 weeks, and we combined those with samples that had been taken in previous years. And we thought that the capacity to understand fine scale changes in allele frequencies would be beneficial for trying to identify the genetic loci underlying allele frequencies. And so having a dense time series would be something that would be beneficial as opposed to having just these primarily these endpoint samples. Okay, so we utilize these samples here in Charlottesville. And we did a bunch of modeling and basically identified that there are a strong enrichment of SNPs that are associated with maximum temperature in the 2 weeks prior to sampling. And this is a sort of a particularly strong enrichment on chromosome 2L. This sort of enrichment is much more than we would expect by chance. And we built a bunch of different environmental models. We built about 100 different environmental models, thinking about different aspects of temperature and rainfall and humidity at different points in time prior to sampling. And this particular model of maximum temperature is the model that clearly beat all of these different ones and clearly beat permutations. We then looked at the distribution of these SNPs across the genome. So these are the 4 autosomal chromosomal arms in flies. And the y-axis is sort of a composite measure of how much signal there is in a window of the genome across multiple SNPs. And what immediately jumped out to us was that there are a concentration of these SNPs near the inversion IN2LT, and that the concentration of these SNPs near IN2LT vastly exceeded permutations, which is the red line in the background. We've seen a similar pattern to this before. In this previous work, looking at samples around the world, we found that there was an enrichment of seasonal SNPs near the breakpoints of the IN2LT inversion. So it was very exciting to see sort of a replication of a previous result. So what is IN2LT? IN2LT is a 10 megabase paracentric inversion on chromosome 2L. It's like not super young, not very old. It's maybe about 15,000 years old. It originated in Central Africa, like all other, like melanogaster did, and spread around the world as flies migrated around the world over the last 10,000 years or so. It has about 1,500 genes in it, so it's got a lot of stuff going on. IN2LT is interesting in that it is found at frequencies of around 15 to 30% around the world. This is data that combines modern pool-seq estimates of the inversion as well as more classical estimates based off of polytene chromosome squishes. Work that we're looking at now and the stuff that we publish about Charlottesville indicates that it has relatively large allele frequency fluctuations within populations through time. We observed this in the Charlottesville samples. We also observed this in the samples from Odessa, Ukraine, as well as samples from Turkey, where we have pretty dense time series. But interestingly, it has a relatively low level of allele frequency change across spatial gradients. So it's being held at sort of a constant frequency across populations, but is fluctuating in time through those populations. And we're trying to understand what's driving that pattern. IN2LT is not a gigantic linkage block like some of the other inversions that are present in other systems. There are islands of the region inside the inversion that are linked to the breakpoints. And then there's regions inside the inversion that are relatively unlinked. So this is a plot of window-based estimates of R squared. And then down below is looking at the linkage disequilibrium in windows along chromosome 2L to the breakpoints themselves. The yellow lines here represent regions that we'd identified as having a strong excess of seasonal, of temperature sort of associated SNPs. And you can see that those regions are largely linked to the breakpoints. So we wanted to be able to understand what are the phenotypes that are associated with IN2LT. And so we assembled a data set of over 200 published phenotypes for the DGRP strains. These we classified into aspects of behavior, morphology, life history, stress tolerance. And these lines, the DGRP is a population of inbred lines that were collected from a farmer's market in Raleigh, North Carolina, the United States, about 20 years ago. They've been fully genome resequenced and have been distributed around the world for people to use for association studies. They've also been karyotyped for their inversion status, so that information was readily available to us. And we performed a test where we basically just generated a t-test, right, between the phenotype and the inversion status for a bunch of different inversions, IN2LT, as well as cosmopolitan inversions that are found on chromosome 2R, 3L, and 3R. The colored diamonds represent our observations. So this is the number of phenotypes that have a significant association with the inversion at a p-value of less than 0.05. The gray circles behind it are permutations, right? What we would expect by chance. And there's a little asterisk if there's more phenotypes observed in the real sort of ordering of the data than 95% of the permutations. So what I hope you can appreciate is that chromosome IN2LT is associated with a lot of different traits and behaviors, more so than the other inversions that we assessed. We took the traits and behaviors that were associated with IN2LT and we performed a principal component analysis on them. There's a lot of things going on in this figure. The general sort of kind of thing that we took away from it was that there was an association between the inversion and aspects of behavior that were associated with startle and activity, as well as things associated with chemical responses and pesticide tolerance. Next, we wanted to try to understand whether there were any regions of the genome where there was an excess of SNPs that were both associated with IN2LT and associated with temperature change. And so we walked along the chromosome and we identified the SNPs that were associated with a particular phenotype, and we identified the SNPs that were associated with allele frequency change. And then we asked, in that window, are those SNPs overlapping, right, more than we would expect by chance? And then we counted the number of phenotypes for which there was a significant excess, and that's what the y-axis is here. And what you can see is that near the breakpoint of IN2LT, as well as some of the windows we had identified inside IN2LT, there was a strong excess of SNPs that were both associated with phenotype and associated with temperature. And the work that we've been doing since then has been to try to understand the genetic architecture of this inversion, as well as to understand try to get a sense of the loci that are associated with the inversion and some of the additional phenotypes that are associated with the inversion. So we're working to perform fine mapping to identify causal loci using Drosophila genetics. We're working to replicate phenotypic associations in an outbred population to test for shifts in overdominance and shifts in dominance. And ultimately, we want to test hypotheses about epistasis and pleiotropy at IN2LT. And I want to tell you just a little bit about some of the progress we've made on some of these projects. So first, we wanted to be able to test whether this breakpoint region was actually associated in an independent way with startle response, which is one of the phenotypes that we had measured. And to do this, we used an assay called a quantitative complementation using deficiencies. And what we do is we cross flies that are homozygous for either the standard or the inverted karyotype to a stock that is heterozygous for a balancer, which is a special type of fly chromosome, and a deficiency, which is a deletion of the genome in a particular region. And we were testing a deficiency that spanned this breakpoint right here. And in this assay, we found that this deficiency failed to complement. And what that means is that in the deficiency background, there's a strong difference between the inverted and standard karyotypes that's not present in the heterozygous background. And this is evidence that that genetic region uncovers allelic variation between the inverted and standard karyotypes. And we're hypothesizing that there's a vesicular glutamate transporter in that region that seems to show a signal of association. And so we're pursuing that as a candidate gene. We want to be able to understand whether IN2LT shows evidence of overdominance, which would promote the long-term persistence of this polymorphism in natural populations and also potentially explain some of the allele frequency patterns that we observe across the environment or across the range. And we also want to test whether there's shifts in dominance across environments. Shifts in dominance, whether the inverted or the standard allele is dominant, has important implications for seasonal evolution. It promotes the stability of alleles in seasonally fluctuating environments. And so to test this basic question, we generated outbred swarms of flies that were fixed for either the inverted or standard. And then we intercrossed these swarms to generate outbred flies that were either homozygous for the inversion, heterozygous, or homozygous standard. And then we reared these individuals in a fully factorial design, either as larvae at 15 or 25, and then we held individuals at either 15 or 25. And we've been measuring a battery of traits on these different genotypes in these different environments: cold survival, development time, life history traits, resistance to different types of stressors, activity and foraging behavior. Some of this work is still ongoing, so I just want to give you a taste of what we've been up to. This is looking at cold survival. And the assay is to put flies in a saltwater bath that's held at -5°C for 3 hours. So this is a pretty cold environment for Drosophila melanogaster, and then measure survival 24 hours later. So a fairly ambitious experiment, 6,000 flies. And we do, in fact, observe shifts in dominance and overdominance. So just to walk you through this figure, each column represents the 2 different larval treatments, and then the rows represent the 2 different adult treatments. We measured both males and females in this assay. One thing to note is that if you rear flies as larvae and as adults at 15 degrees Celsius, they have a pretty high survival to this cold assay, 80% upwards. Whereas if you rear flies as larvae and adults at 25, they have a very low survival on the order of 10%. And there's an effect of both the larval and the adult environment on this survival. There's also an effect of genotype, right? When you rear flies at 15 degrees C as larvae and adults, the standard allele is dominant. Whereas if you rear flies as larvae and adults at 25 degrees C, the inverted allele is dominant. And at intermediate temperatures, there is a reversal of, there is evidence of overdominance. And so this is meant to indicate that it's possible that the shifts in dominance and signals of overdominance could contribute to the allele frequency dynamics that we observe in the wild. In general, we observe that there have been shifts in dominance for some of these other phenotypes, a little bit less evidence of overdominance, but we're still working through some of these analyses. Okay, so what are the loci and phenotypes underlying seasonal adaptation? Well, INTOLT seems to be a locus that fluctuates, and it seems to be a major seasonal locus associated with dozens of behavioral stress tolerance and life history traits. And there is potential evidence for overdominance or marginal overdominance, which is caused by these shifts in dominance across environments. I want to wrap up just by talking about why I think this work is important in general. So one, I think that it shows that fluctuating selection is an important evolutionary force. The second is that adaptive tracking is likely a common phenomenon, right, at least for short-lived organisms. And finally, I think this work is interesting because it highlights evolution in action, right? So when I talk to people about the work that I do, especially when they're not scientists, the first question I always get is asking me, how can I get rid of these things in my kitchen? So this is a picture of flies in my kitchen on a peach. And so in response to this question that I was getting, we started a project looking at the evolution of flies in people's backyards. And we started what we call the Backyard Evolution Project, where we work with local gardeners to sample flies from their backyard compost piles throughout the growing season. So this is a project that we've had going on for now 4 years. We've collected over 50,000 flies from over 30 collectors and sorted them down to about a dozen species. We are trying to use this data set in conjunction with samples we've collected at orchards in the same region to ask questions about microspatial variation and selection pressures between orchards and compost piles. At least out in Virginia, orchards spray pretty heavily because if they don't, the fruit gets infested with different types of pests. And flies respond to those pesticide selective pressures. It's unlikely that compost piles in people's backyards have the same sort of selective pressures. And so we're trying to use this information to test hypotheses about local adaptation, potentially related to human activity, as well as local adaptation to different types of environments, right? Compost piles are would be a very terrifying place for a larval Drosophila to live or for an adult Drosophila to live because of the large numbers of predators and competitors, whereas in an orchard, it's a very resource-rich environment and melanogaster is very efficient at exploiting fermenting fruit. With this work, I would like to thank the members of my lab that contributed to it, as well as my collaborators and funding around the world. And with that, I would like to take any questions.
Thank you very much. Do we have questions online? I don't see. No, we don't have questions online. And in the audience, we have question.
Yeah, thank you very much. Since this is school, is it school? Yeah, this is school, and silly questions are allowed. I have a silly question. So you have an inversion, so chunk of DNA just changing direction. Why it affects translation of all the genes which are on this inversion chunk?
Yeah, so the question is, how many genes does this inversion affect the function of, either maybe the transcription or the translation or the function of those different proteins? That's a really good question. There's been work looking at differences in expression between the inversion and the standard genotype. And what has been found is that the inversion itself doesn't seem to disrupt the expression of the genes that are right at the inversion breakpoints. It does affect gene expression of genes in the inversion, but not any more than affecting the expression of genes outside the inversion. So there's very large sort of pleiotropic effects of the inversion itself. And what are the causal genes that are basically associated with the inversion and its phenotypic effects and adaptive value are unclear. There are a handful of regions inside of the genome that are very tightly linked to the breakpoints. And so we hypothesize that those could be affecting it. And those particular genes, we don't have any indication of how, though. So that's something that we're working on. And yeah, it certainly does not affect every gene in the inverted region. And also there's the suppression of linkage extends outside of the breakpoints. And so there's also genes outside of the breakpoints that are pretty tightly linked to the inversion, and it could be those as well.
Do we have other questions? So I have question. You analyzed huge database which include samples from populations where Drosophila have diapause and where Drosophila doesn't have diapause. Did you see any difference in seasonal adaptation patterns, maybe in genes or allele frequencies of some genes in such two types of populations?
So the two populations are what again? I'm sorry, I missed it.
But you analyzed DEST too.
Yeah, can you, I missed the two different habitats that you were asking about. Could you repeat?
Yes, in some populations in this data set, Drosophila have diapause. In some populations, no, not. Is there a difference of allele frequencies of some genes, or do you have the same picture in such two types of populations?
Yeah, so the question I think is about populations that have diapause and populations that don't have diapause, and do we see shifts in allele frequencies in these populations? So that's a really interesting question. We haven't looked at it exactly like that because we don't know too much about the genetics of diapause. We did a study years ago looking at, we did an association study looking at loci associated with diapause variation. And we were then able to take the SNPs that were associated with diapause variation and look to see if they change in allele frequencies in the way that we would expect, like spring is high diapause, fall is low diapause. And what was very interesting was that we observed that in some populations, there was sort of a predictable change in allele frequencies, but in other populations, the change in diapause allele frequency went in the opposite way from what we would expect. But that could be because we just didn't quite understand what spring and fall were in those samples. Did that answer your question?
Thank you. Yes. And we have one question online. Valerie, please.
Thank you for amazing presentation. I would like to hear more about citizen science part. So you said that starting from the question, like from someone, how do I get rid of those flies? You like proposed to collect flies in different places, and if I remembered correctly, like 30 people participated. So what did they get? Like, did you tell them how to get rid of them, or maybe like you have some app? Because I do bird watching and I use some apps, and I just like to collect information about birds to do this list. And I know that scientists analyze this data, so it's like amazing example of citizen science bird watching. And I just want to hear more about your approach.
Yeah, cool. Thanks for the question. Right. So I guess one place to start is to talk about the ways that we have tried to recruit people, and then to talk about kind of what they get out of it. We've started to we've primarily recruited through an organization in the United States called the Master Gardeners. And the Master Gardeners organization is organized through the agricultural universities in the United States, and they're people who like to garden, and they get together every month and hear presentations about like how to grow better peaches or lettuce or whatever. And these are just like backyard home scale gardeners. But part of their sort of responsibility as being a master gardener is that and in order to maintain their status as a master gardener, they need to volunteer in different ways. And so a lot of them do outreach at farmers markets where they just sort of talk about gardening and try to promote gardening in the community. And so I've gone to their clubs to present about the work that we do and to talk about the importance of model organism science and evolution. And so, you know, these people aren't scientists, and so they appreciate, I think, hearing just sort of about what science is about and what evolutionary biology is about. And then so I've recruited from there. We've gone with master gardeners to the farmers markets with them and brought our microscopes and like buckets of compost. And then like kids and adults can look at compost on our microscope, which if you've never done that is an incredible experience because it's just teeming with life. And so we've recruited volunteers through these farmers market outreach efforts. We send volunteers these kits, and so this is our kit. It includes a Drosophila net and collection vials. We send out reminders every 2 weeks and postage to send samples back to us. We have had about 30 successful collectors, but there's been about maybe 75 people that we've sent kits out to. A lot of people don't have compost piles that are conducive to fruit flies. So you really need to be like throw a lot of fruit and vegetables and also be kind of lazy. Like don't turn your compost pile a lot. If you put a lot of leaves and grass and things like that, then it becomes hard to collect flies from because they don't like it there. So we've learned a lot. One thing that we're sort of yet to do is to really like let all of our volunteers understand the results of our work. And that's just because like the results are still sort of coming along, but I look forward to in a year or so organizing a small retreat or symposium online to be able to invite these people.
Sounds awesome. Thank you. And I will watch compost under my microscope. Sounds interesting.
Looks like there's another question online.
Another question online, Tatiana.
Thank you very much for this interesting report. I just like to know how does such chromosomes with inversion, how do they survive crossing over? Is there any information about this available?
Yeah, right. So how do they survive crossing over? Well, I think the short answer, my understanding is that if there's a single crossover event between the inverted and the standard, the recombinant offspring don't survive because they have large duplications or deletions that create problems, right? So there's a suppression of recombination because of that unequal crossing over, essentially. But the inversion is large enough and old enough and recombination rates are high enough in flies that you can get multiple recombination events on a chromosome. And if there are double recombination events, then you can actually get recombination between the inverted and the standard. And so that's what basically causes this inversion to not be a gigantic linkage block, but instead to be a sort of have signals of high and low differentiation.
Oh, thank you.
Do
I
we have
see.
questions from the audience? Oh, I think no. Thank you, Alan, for your great presentation.
Okay, thank you very much for the invitation. Let's stop sharing my screen.
Thank you.
Yeah, I'm trying to figure out how to get my thing back. 1 second.
And now it's my pleasure to introduce our next speaker, Andrea Bettencourt. She started her education at the University of Texas, and she completed her PhD at the University of Rochester. And did her postdoc at University of Edinburgh and in Bedmonduni, Vienna. And now she is she's working at the University of Liverpool, and she is an evolutionary biologist working to answer questions about adaptation, such as do sex and recombination allow rapid evolution? Does sex linkage promote adaptation? And how organisms adapt so rapidly to transposable element invasions? We are happy to start this lecture. Andrea, the floor is yours.
Okay, thank you very much for that introduction and for the invitation to come talk to you guys. I'm talking to you from Liverpool, which you probably know is famous for football and for the Beatles, but which also hosted Eurovision on behalf of Ukraine. So it was really nice to see all of the activities to do with Ukraine that came up along with this hosting of Eurovision, where people tried to celebrate Ukrainian culture in Liverpool. I'm here today, like the last speaker, to talk to you about my work in flies. This work is particularly to do with transposable elements. And if you don't know what transposable elements are, I will try to quickly explain. They're basically just selfish little pieces of DNA that are like other pieces of DNA inherited from parent to offspring, but which also replicate within genomes, increasing copy numbers. These pieces of DNA, these selfish genes, are really successful. They tend to make up a lot of the bulk of genomes. So using the human genome as an example, almost half of the human genome is made up of DNA that's derived from these transposable elements, the TEs. You can contrast that with what we think of as our genes, the part that, the kinds of DNA that encode for proteins, and that is only about 1.5%. In fact, TEs are a big explainer of genome size evolution. So this is genome size for different kinds of insects, and you can see as the genome size increases, the transposable element content also increases. So these TEs are making up big chunks of our genomes, especially for eukaryotes, but they may not actually be doing anything beneficial for us. The whole explanation for why they exist and why there are so many of them may just be due to the fact that they can selfishly replicate within genomes. And this is the idea of the selfish gene, popularized by Dawkins, but also brought up in the scientific literature. I particularly like, if you're just starting out, this Lewontin paper about units of selection, which really gives you a perspective for all the different kinds of ways in which selection can act, not just at the individual level, but also within individuals. Okay. So, as I told you, TEs can spread within genomes by selfish replication. They can also make up large proportions of eukaryotic genomes. One thing that's probably been underappreciated until quite recently, that as transposable elements jump, they can cause mutations. Like other kinds of mutations, these mutations are usually harmful, but they're sometimes beneficial. So you can see this by looking at some of the most famous mutations. You've probably encountered this story about the melanic peppered moth that spread along with industrialization in most famously over here in the northwest of England, where the Industrial Revolution got started, particularly in Manchester, but also throughout Europe and throughout America. You probably also know this mutation, which is the wrinkly P mutation that Mendel studied. That also appears to have been a transposable element. And you may not know this mutation, but I work on flies and I find this mutation is very famous. This is the very first white allele, white 1, that was isolated in the Morgan lab back in the beginning of the 20th century when they were just starting to study Drosophila genetics. And that is, again, due to a transposable element derived mutation. Like other mutations, these transposable elements sometimes cause beneficial effects. These seem to be particularly important in domestication. So there's a famous case that's probably led to the formation of nice ears of maize or corn from the sort of spread out seed head of its ancestral species. Insecticide resistance is where another case where transposable element derived mutations seem to be important. One really cool story is there is a transposable element gene that's been co-opted to use in mammals at the placenta fetal interface, the syncytia. And that's true, that co-option of that particular gene, the use of that particular gene has happened a few different times independently in mammals and also independently in lizards that have this placenta-like structure. So I think that's a really cool story. Okay, there we go. So just wanted to quickly introduce you to transposable elements. They are selfish genes, make up large parts of our genomes. As they jump, they cause mutations. Like other mutations, they're mostly harmful, sometimes beneficial. But importantly, because they're mostly harmful, it's important to understand that the host very quickly evolved to suppress these transposable elements. This suppression causes the TEs, I'll probably start calling them TEs because it's a lot shorter to say than transposable elements, so please don't be confused by that. So as these TEs are mostly harmful, hosts quickly evolve to suppress them, and when they're suppressed, they lose most of their ability to replicate selfishly, and then they start to degrade in genomes in place. This, if the hosts are quickly evolving to suppress them, it raises the question of how they're maintained in the long term. So one clue has come from phylogenetics, where you can see that the transposable elements, you can make a phylogenetic tree for the transposable elements. You can make a phylogenetic tree out of the host genomes. And you can see that these two things don't always match. And sometimes, for example, very closely related host species have very different transposable elements, and sometimes very similar transposable elements appear in very different host species. This appearance of very different transposable elements in different host species suggests that the transposable element is being horizontally transferred like a virus between host species. So this idea, this sort of model has come up that transposable elements are partly surviving by horizontal transfer. So to show you how this works, you have a horizontal transfer event, and then the transposable element is unsuppressed and can increase and replicate within genomes. And then the host evolves suppression, and that can lead to rapid decay. So in that case, the transposable element has to escape and invade a new species. This kind of dynamic can also happen just via escape of suppression. If the transposable element evolves enough in sequence, it can escape host suppression and start to proliferate again. So that's another way they can escape host silencing is either through horizontal transfer or through evolution of their sequence. Okay, so this starts to get a little bit historical because we didn't always know about transposable elements. Eukaryotic transposable elements in particular were studied very early on by Barbara McClintock, who was the person who discovered transposable elements back in the 1950s or so. And then there was a long period of hiatus of studying eukaryotic transposable elements. Mostly, people knew about them and studied them in bacteria, but they were mostly neglected in eukaryotes. They were rediscovered again by discovering this transposable element called the P element. The P element was found actually in Texas, where I'm from, because it affected reciprocal crosses differently. So this is a phenomenon called hybrid dysgenesis, and hybrid dysgenesis affects reciprocal crosses differently. What does that mean? The observation was that if you take a wild-caught male and you cross this wild-caught male to a female that's derived from a lab strain, very often the offspring of those two end up being sterile. This is a picture of the ovaries of a female from a cross such as that, and those ovaries are completely degenerated. If you do the cross in the opposite direction with a wild-caught female and a lab male, instead the females are perfectly fine. The offspring are perfectly fine. They have, in this case, nice, fat, full ovaries. Okay, so this is very strange because these two flies are genetically identical. They have identical nuclear genotypes, but they have very different phenotypes. This is a nice, normal, fertile female, and this is a infertile, sterile female. Through lots and lots of genetic work by molecular biologists and other geneticists, people figured out that this difference in phenotype was due to a 2.9 kb DNA transposon, a kind of transposable element that exists in the wild flies but was absent from the lab flies. This slide is cut off, sorry. This really was an important discovery for biotechnology because this is how transgenics was done in Drosophila for many, many years. And lots of different transgenic tools in other organisms are based on the same basic idea by taking a transposable element and inserting it into the genome, along with whatever DNA you want to also insert in the genome. Another thing that was very interesting about this early study of a P element is that it was clear very early on that it was quite similar in sequence to a transposable element in this species called Willistoni. So Willistoni is also a Drosophila species like Melanogaster, the flies we're talking about, but it's very distantly related. So this is about 65 million years of evolution that have happened between Willistoni and Melanogaster. Nevertheless, the P element that those two species have is really similar in sequence. There's a one base pair difference. The rest of the genome is much more different, but the P element appears to be almost the same, which strongly indicates that it was horizontally transferred from Willistoni into Melanogaster. Another piece of evidence that the P element was horizontally transferred is that you can find it in Drosophila melanogaster, the flies where it was discovered, but it appears to be mostly absent from close relatives, or completely absent from close relatives. So this is Drosophila simulans, if you know that species, and some other relatives of melanogaster. Okay, so once P. element appeared in Drosophila melanogaster. It appeared in the Americas, of course, and it spread rapidly there. Here you can see very early on in the spread, it's already spreading through Europe. So these are these dark circles here. And the open circles also have some evidence of P. element. So these things are spreading quite rapidly worldwide. So this is, as an evolutionary geneticist, I'm really interested in these kinds of phenomenon because they're not really how we think of, or how I used to think of evolutionary change. I'm used to thinking of evolutionary change as you have a sequence, there's a change in that sequence, and it increases or decreases in frequency. And it, you know, many years in the future, you could end up with a different genetics in an organism. This, though, is genetic change happening almost in historical time, where you have, you know, Morgan collecting his flies in the early 20th century. You have me collecting flies many years later. This is in Vienna. And some of the biggest differences are that they harbor different transposable elements acquired by horizontal transfer. And they also have a different Wolbachia that has spread through melanogaster. These are both things that Sveti also works on. Okay, so the P. element spread in Drosophila melanogaster. However, that was not the end of the story. That became clear when I had my first PhD student. We started doing some crosses. We did some crosses not in Drosophila melanogaster, but in Drosophila simulans. And these were just some flies we had around that came from very different geographic areas, and we wanted to see if they showed any hybrid dysgenesis. And so they're flies from Georgia in the US and from Madagascar off the coast of Africa. So this is my first PhD student. He did lots and lots of crosses, collected an amazing amount of data, and he found, just like in melanogaster with P. elements, when you cross, in this case, instead of a wild male, a male from Georgia, to a female from melanogaster, you can see this sterility phenotype. And of course, when you do, it's important to show that when you do the reciprocal cross, you don't see that phenotype. So the reciprocal cross with the genetically identical flies is normal. Okay, so we saw that lots of Georgia males, this data show that lots of Georgia males caused hybrid dysgenesis regardless of which Madagascar female you cross them to. So we're seeing repeated hybrid dysgenesis in these flies induced by Georgia fathers. We were then able to look at the transposable elements in these flies and ask which ones were associated with this dysgenesis phenotype. And when we did that, we found that, much to our surprise, the P. element was there. It was not supposed to be in Drosophila simulans, and less surprisingly, is highly associated with this hybrid dysgenesis phenotype in simulans as in melanogaster. Okay. We also looked at the spread of the P. element in Drosophila simulans. So I have to say thank you to DROS-EU, which both Alan and Sveta are involved in as well, because through DROS-EU, I was able to ask people for their collections of flies, and they were very generously sent me flies that they had collected from all over the world. So we did this. We just checked for P. element with PCR in different populations, and you can see that the P. element, which is the part of these circles in red, is starting to pop up around 2008. There's this one sample kindly donated by Evolutionary, which is a very large sample collected in Portugal, where we first see P. element in 2006. Importantly, also, these flies were turned into DNA in 2006. Most of the rest of these are living cultures that are propagated in the lab, and you can imagine that maybe they used to have P. element but lost it, but you can't say that about these samples because they were DNA since 2006. Okay. If you look later on, flies collected in the wild after 2009, you can see that they have you can see much more red on this map, where you see more and more red filled in circles, and pretty much by 2012, all of the samples we find have at least one P element in them. Okay, so the P element briefly hopped from Drosophila willow-stoni to Drosophila melanogaster to Drosophila simulans. A question I always get is, how did this happen? And I'm afraid the answer is, I don't know. I wish I did. There are lots of hypotheses out there, but for the moment, it's just easier to say, don't know. Okay, so we continued working on the system, looking at the suppression and looking at other things. One of the projects we've done is looking at dysgenesis phenotype. So we have the dysgenesis phenotype in males. The main one we use is this ovary phenotype, but there are others. One thing we wanted to know is what's going on in the males, and does the P element, for example, damage male fertility? So this is known in Drosophila melanogaster that there are some interesting phenotypes associated with dysgenesis in males, but we wanted to look at this in females as well. And this is work led by postdocs Joanne and Valeria Romero-Soriano, who did this work and did the analyses, except for one. I did one analysis, which I'm very proud of. I don't get to do analyses that much these days, and so I really, really value it when I can. Okay, so dysgenesis, like in Drosophila melanogaster, affects reciprocal crosses in Drosophila simulans differently, as I told you before. So we have P element containing strains. When they're the female, the offspring are normal. When they're the male, the offspring are sterile. So what's really going on there, is that they are inheriting different cytotypes from the female parents. So the P element female passes on the P cytotype. You can think of this as kind of the egg cytoplasm. And the females that lack the P element pass on something called an M cytotype that doesn't protect the offspring from this sterility phenotype. We know now from work from molecular biologists that what's going on here is that there are small RNAs that act to protect the germline from transposable element activity, not just the P element, but many other kinds of transposable elements as well. And these pi RNAs are loaded into the cytoplasm by the mother and then protect the offspring. When the mother has the P element, she can load pi RNAs that protect her offspring from the P element into her cytoplasm. And when she lacks the P element, she lacks those pi RNAs, and therefore her offspring are unprotected and can be sterile. Okay. So we wanted to look at males, and the first problem we have is that if you make reciprocal crosses, the females are genetically identical, but the males differ in their sex chromosomes. So the normal situation would be that you could make a hybrid dysgenic cross using a female that lacked the P element, and a reciprocal cross using a female that has the P element. And they should pass on their cytotypes and protect their offspring in the case of the P element containing female, or not be able to pass on P element protective pi RNAs against the P element to their offspring in the case of the female that lacks the P element. So these two males are from reciprocal crosses. They differ in their cytotype, and they also differ in their sex chromosomes. This X chromosome comes from the the female that lacks P element, and this X chromosome comes from the female that has P element. How do we get around that? We use fun Drosophila genetics in the form of an attached X chromosome. So this is a tool that exists in simulins and in many different copies in melanogaster as well. This allows you to get genetically identical reciprocal males. So now this male has the X chromosome from the P-containing father and the Y chromosome from the P-lacking mother. And this male has the same sex chromosomes. However, they differ in their cytotype. If you're interested, Drosophila genetics is really cool. How this works is the two X chromosomes of the mother are physically attached and segregate together. When this happens, and so in this cross with an attached X female, males actually inherit their X chromosome from their father and their Y chromosome from their mother. So the opposite of the usual situation. Okay, so we're able to get these genetically identical males that have identical nuclear genomes but differ in their cytotypes. When we compare these two males, so now we have comparable males, we can see that the dysgenic males suffer infertility compared to the non-dysgenic reciprocal males. So this is their total offspring production over their lifespan. You can see it increases and then decreases. Early on, there's very little difference, but as the males age, the dysgenic males suffer lack of offspring production. It's not quite the dramatic sterility you see in the females, but it is lower offspring production compared to reciprocal males. We also did some RNA-seq and looked at transposable element expression in the testes. There were a couple of differentially expressed transposable elements. The main differentially expressed transposable element, however, was the P element, as we expected. So that was nice to see. They also showed, dysgenic males also showed a general kind of phenotype that suggests a lot of dysregulation is possibly going on. So there was some dramatic misregulation of proteolysis genes and other kinds of metabolic genes, which suggests something funny is going on, which would not be surprising given what's happening in the female germline. The male germline may also be suffering from misregulation. Okay. Another thing we wanted to know, which was a little bit harder to find out, is if P elements are mobilized in males. One idea we had early on was that maybe transposable elements are really well regulated in females and so spreading more in males. We looked at transposition directly in both males and females, and we did this by comparing new insertions in the offspring of dysgenic and reciprocal F1 flies. So we look at the F2 to see what transpositions happened in the germline of their parents. We compared dysgenic and reciprocal F1 males and dysgenic and reciprocal F1 females. Unfortunately, we can't really directly compare males and females because we were unable to get genetically identical males and females. Okay, then we identified these new insertions. We did this by using targeted PCR, some clever pooling, and some barcoding to screen insertions so that we were able to screen over 2,000 flies each. So we did this, and we found that we indeed saw more new insertions in F1 females if they're from the hybrid dysgenic direction of the cross, where we also saw the higher P element expression. And we also saw more insertions in F1 males. So both in hybrid dysgenic F1 males. So in both males and females, the lack of suppression leads to more P element mobilization. So the offspring of these hybridogenic males have more copies of the P element. Okay. So that's a story about the P element, which is one of the first ways we came to think about horizontal transfer and proliferation and suppression and decay of all these transposable elements. But of course, the P element isn't the only game in town, and Drosophila isn't the only organism out there. So in my lab, especially with this fabulous PhD student, Flora Todd, we started to look at other species and other invasions of selfish genes. So this is a collaboration with my colleague Ilik Zakiri, who is responsible for finding out that the genetic basis of the peppered moth, the melanic form of the peppered moth that spread with industrialization was due to transposable elements. So he's continued to collect a lot of genomic data on Lepidoptera, and he's very kindly letting us use it to understand transposable element invasions. So as I said, the P element wasn't the end of the story. In fact, even in Drosophila melanogaster, we know that there have been other invasions that have happened in historical time. This comes from recent work, even more recent work showing, that came out just last month, showing more invasions. The situation in Lepidoptera, if anything, ought to be more dramatic because Lepidoptera in general appears to experience more horizontal transfers than other kinds of orders of insects. So these are different orders of insects. And if they all had the same rate of horizontal transfer, they'd all appear on this line. You can see that Lepidoptera is rather dramatically above that line, so appears to have even higher rates of horizontal transfer. Okay. So we started by looking at the peppered moth data. So this is Biston betullaria. And we were able to do this because there's lots of high quality genome sequence data, including that has been collected in Ilik Zakiri's lab, including 5 PacBio hi-fi generated genomes that are fairly complete, and short read data from 600 individuals that he went out and trapped mainly in Europe, and another 250 museum individuals that he was able to get permission to sample and collect short read data from. These museum individuals were collected in the early to mid 20th century. And of course, what we're going to try to do is find differences between these museum and modern individuals that suggest transposable elements that have invaded between the time the museum individuals were collected and today. And this is Ilik looking very happy, and his fabulous postdoc Sam, who's also been very helpful with us understanding and using these data. Okay, so we're going to try to look in the sequence to find transposable elements that are different between museum and modern samples. And one of the things we have to ask ourselves is, do we really trust the sequence data? We have to use, we're going to look at coverage of the different samples to identify transposable elements that are covered by the sequence data in the modern individuals and not covered by the sequence data in the museum individuals. So this led to us to wonder if we could really trust the coverage in those museum samples, because the sequences are, the coverage is lower, the sequences are a bit more degraded. It's a little bit more difficult to just take at face value a lack of coverage from the museum samples. So what we did is we went and looked at something called busco genes, which are genes that you ought to find in your samples, and they ought to be a single copy. And we looked at, we just used data from RNA-seq methods. So RNA-seq is a way of looking at gene expression through coverage. We just want to look at presence and absence through coverage, so we just use those RNA-seq methods. to get normalized coverage of these single copy genes in the modern and museum samples. And I hope you can see that they're pretty well correlated. There's variation here due to gene length, but in general, if something has high coverage in the modern sample, it usually has high coverage in the museum sample as well. So we think we can start to trust the museum data, although of course we have to keep being careful. So this is ongoing work. We haven't finished it, but we hope to finish it soon. We're using two different methods to find transposable element invasions. First, we're taking reads and mapping them to a TE library. So this is just a list of sequences that we've curated from the BISTON reference genome to look for things that we think are transposable elements and ask if they're covered in the museum sample, in the modern sample, if reads map to those possible transposable element sequences in the modern samples and don't map in the museum samples. However, this relies on us capturing all the interesting transposable elements in a TE library. That has turned out to be really challenging, much more challenging than I had thought it would be to start with. However, so we also are going to look at just naively differences in coverage between the museum and modern sample of the reference genome, looking for regions that are not covered in museum samples or show lower coverage in museum samples, but are covered in the reference genome. Okay. Oof. What have I done? Okay. I won't go into great detail about this TE library curation, other than to say it's been a lot of work, mostly for Flora, and it's, I'm really grateful that she was able and had the patience and tolerance to go through all of these sequences. So I think in the case of BISTON, I think she had to look at 1,500 sequences in order to manually curate these libraries, which is an enormous amount of work. Okay, so far, what have we found? So we're still looking, but so far we have found some TEs that are higher frequencies in the modern compared to museum. So you can see this as an example here. We have a transposable element consensus sequence and the reads that are mapped to it. This is an example of an individual that has reads mapping all along this transposable element sequence. And here's an example of two different individuals that mostly don't have reads mapping to this transposable element sequence. These regions at the end are regions that probably don't actually belong to the transposable element. Also importantly, when we blast and look for this transposable element in close relatives, it appears to be absent. We look at so we look at this spread through time, and it does appear to be spreading. These remaining bits of sequences are those in bits. So these probably correspond to 0 coverage, and this to higher, to actual presence of the transposable element. Hopefully you can see already the importance of manual curation. We need to cut those ends off. This was also emphasized to us with a different transposable element. So we had a transposable element that was automatically identified, and we'll just call it TE2. This transposable element, when we looked at it carefully, appears to be 2 different transposable elements that have been clumped together. And it turns out that they're clumped together because the 3' end, I should say 5', sorry. The 3' end of the transposable element is nearly identical, and the 5' end is unique. once we separate those two, or once Flora separated those two with manual curation, you can see that one of these transposable elements is present in all of the samples, and the other one appears to be invading through time. So this transposable element, which we've just called TE2A, is absent from some of the museum individuals and seems to increase in copy number with as we go through time towards the present day. Okay, and again, this TE is absent from close relatives of this species. And in fact, when I say absent from close relatives, absent from almost anything closely related. The last thing I wanted to talk about, and I think it's important to show that, that this transposable element looked pretty flat, so we can find transposable elements that don't look like they're spreading. That's important to show because we're going to look at the transposable element that appears to be responsible for this difference in peppered moth phenotype. So this is a TE that was named in this paper as carb TE because it's responsible for this carbonaria form of Biston betullaria. And this carb TE is only in 4 copies in the Biston reference genome. So the Biston reference genome is from the UK. In these European Biston from the continent, however, it appears to be in many more copies. So we estimate copy number to be around 50 for most of these samples. It also appears to be spreading through time, getting a little bit more common in present day samples than it was in the past. Okay. So that is telling you about transposable element invasions in Lepidoptera. We think we've captured 2 ongoing invasions so far. We're still looking, and these happened quite recently in historical time. We also think the carbonaria TE may be increasing in frequency, and hopefully I've convinced you that TE invasions are interesting and can happen almost as we watch them. I have lots of people to thank, particularly Flora, Elix Carey. Carl Young is the technician who did some really amazing molecular biology to get those museum samples to actually be much higher quality than I've seen before. I'd also like to thank my lab, including Joanne and Adriana, whose work I didn't have a chance to talk about, but she's a really fabulous bioinformatician in my lab. And lab technicians and Joanne for doing analysis and lab work to do with the transposable elements, dysgenesis phenotype in Zimmermans. Okay. I'd love to take questions. I've been talking to you guys a lot today, so sorry, my voice is starting to go.
Thank you, Andrea.
Thank you, Sveta.
Do we have questions online? Well, for now, I don't see questions online. And do we have questions in the audience? Okay, we have questions.
Okay, so just please hold a while, I'll take my notes. So, okay, so first of all, you were talking about transposable elements and its influence on genome, and especially like I'm interested in its application on humans because first of all, we have many diseases that are induced by some mutations. So, I mean, my main question is that can we apply gene editing tools like CRISPR or probably viral vectors to treat gene mutation caused by transposable elements, or maybe are some differences between gene therapy for like usual mutations, maybe caused by deletion inversions or like just mutations caused by transposable elements. So do we have difference in treatment?
So this is a really good question, and I will say it's not my area of expertise, but I'll try the best I can. It is not so getting to a place where you can implement treatment takes a really long time, and we haven't until maybe 10 years ago or so really thought that hard about the role of transposable elements in human disease. Well, at least interest has become much more apparent recently, looking at the role of transposable elements in disease. I know of work looking at when you map genetic diseases in humans, there is an enrichment of transposable element insertions at certain sites in humans. There are associations with cancer. There's thought to be perhaps some associations with schizophrenia. And there are other uses which are less obvious. For example, one thing that people working in human disease really spend a lot of time doing is trying to find biomarkers of disease. So just asking, is there a diagnostic test? And recently I heard a really nice talk from somebody in the US, can't remember where, maybe Tulane, who was using expression of a transposable element in the blood to identify different cancers. And that was quite interesting. As far as using CRISPR, I know you can knock out transposable elements with CRISPR, but as far as I know, this is, mainly done in cells rather than people. There's a question about, is there a pattern where TEs insert in the genome? Should I answer that one?
Yeah.
It's a good question. And there is, although I've been talking about TEs very generically, there's actually in Drosophila, there's a good 100 families. In humans, there's 4 that are active, that are inserting, or potentially inserting. And each transposable element, or at least each kind of transposable element, likes to have its own particular sort of insertion site. So perhaps some insert randomly. The P element likes to insert at origins of replication or near the near transcription start sites of genes. There are particular elements that like to insert particularly into the ribosomal DNA. So that's a very common strategy. And there are small sequence motifs that are associated with transposable element insertions as well. So they do have their own particular places they like to insert, but different transposable elements like different places. Another question. And this question is, in the case of invasion by horizontal transfer, what kind of vector is used to invade the host? I wish I knew. So some speculation has been out there for a long time that it's parasites or ectoparasites like mites, or possibly parasitoid wasps, or potentially Wolbachia. There are large DNA viruses that tend to carry around pieces of DNA. That's a really good candidate. Unfortunately, it's really hard to answer. I mean, you almost need a time machine to answer the question about the P elements. There is some nice work showing a particular transmission of BOV-B, which is bovine B something, transposable element, where you've sort of been able to reconstruct the whole train of transmission from one host to a vector to another host. But in general, we don't really know. We can guess, but we would really like to know. I would especially really like to know.
Maybe one more question in the audience.
Andrea, could you please comment if such invasions happen, even in spite of hybrid dysgenesis, these elements might give some useful thing to the host. Yeah, if this
Yeah.
environment will happen in the population.
Definitely, they do sometimes. So there is some really nice work from several different labs, among them Pepe Gonzalez, looking at transposable elements landing near genes and changing the regulation of those genes. There are cases in particular where this has happened for insecticide resistance. That's a big one, including the P elements. It has landed near a gene, changed the regulation of that gene, and now the flies are resistant to, I've forgotten which insecticide, sorry, but they're resistant to a particular pesticide. I quite like the idea that transposable elements can lead to big changes in fitness, basically. Most of those big changes in fitness will be bad, but every now and again you'll get a change in fitness that is good. Sorry, I'm just trying to think of some more examples, but there are loads of examples where transposable elements have given some sort of benefit to the host.
Thank you.
And this is something we didn't really think about until pretty recently, and now it's all over the place. Like once you start looking, it's one of those things you start seeing everywhere.
Any other questions? Yes, we
No,
have-
it's getting quite late there, isn't it? Yeah.
I think that's it. Thank you very much, Andrea, for your presentation. It was great.
Thank you so much for the invitation and for organizing this.
Okay, so it was last presentation for today, and this day is closed. And then we will have dinner at the same place.